Good day, everyone. I am Alminda Magbalo Fernandez from the College of Agriculture, the Rizal Memorial Colleges Incorporated. At this point, I would like to present to you the result of our study entitled Growth Performance of Tissue Cultured Cavendish Banana, also known scientifically as Musa acuminata plantlets, as affected by stimulate, a foliar stimulans and enhancer for plants. I would like to acknowledge also my companions in this research, Mr. Louis Ray D. Montesilio of the University of Southeastern Philippines, Tagamabini campus, and Mr. John Paul Matuguines in the same College of Agriculture, Rizal Memorial Colleges. So for the introduction, we all know that banana, the Musa species, is native to tropical region of Southeast Asia and Australia, and it is in fact the most important crop in the Philippines in terms of hectare and commercial value. It ranks first in production and area planted to fruits, a major dollar earner with about $100 million annually contribution to our economy. Philippines is also one of the top five producers of banana in the world, and exported bananas are definitely fresh Cavendish, which accounted around 92% of the total export. So, in quest for more alternative growth enhancers, a promising product like Stimulate from Stoller Philippines was tested, hence, this study was conducted. Stimulate is a combination of three different phytohormones known as cytokinin, gibberellic acid, and an auxin in the form of indole-3-butyric acid. These are all necessary to stimulate cell division, cell differentiation, and cell arrangement in all positions of the plant, hence enhancing growth and development of plants. So for the methods, after 6 months from the tissue culture laboratory of the University of Southeastern Philippines, these tissue cultured banana plantlets were transferred into the nursery at the same experimental area of the University of Southeastern Philippines at Tagumabini campus. And they were transplanted using 4 by 6 inches polyethylene bags with 80% coco coir dust and 20% vermicast, and then applied with conventional practice of fertilizer application with amount of 5 grams complete fertilizer after 14 days from transplanting and 7 days or weekly thereafter. So with standard cultural practices in the nursery like watering, weeding, and fungicide and pesticide, insecticide application whenever necessary, and also drainage were all done to maintain and care the plants. And the different treatments were also applied. So for the experiment, we have treatment one, control or without application at all, and we have all the treatment two, which is under conventional practice. That means that we only applied 5 milligrams of complete fertilizer 14 14 14 at as I have said 14 days after transplanting and every week thereafter. And we also have treatment 3, the stimulate growth enhancer at the rate of 10 ml per liter of water, and were applied in as foliar fertilizer every week. And we also have treatment for the stimulate plus in combination with the conventional practice, which is 5 milligrams of complete fertilizer and 10 ml per liter of water were sprayed and applied into the plants. And treatment 5, we have Bioforge at the rate of 5 ml per liter of water, and also conventional practice or the 5 milligrams of complete fertilizer were applied weekly. And we have the treatment 6, the combination of Stimulate and Bioforge alone, with again 10 ml of um, Stimulate per liter of water and 5 ml Bioforge per liter of water. And the last treatment is the combination of the three: conventional practice, 5 milligrams of complete fertilizer, and Stimulate 10 ml per liter of water, and Bioforge at 5 ml per liter were sprayed into the plants weekly. So that there were 7 treatments, and they are replicated 3 times, and with about 10 plants per replication, sample plants. So the study work was conducted for 2 months duration, and after 15 days, the growth parameters were gathered, starting with the percentage survival. All the live plants were counted after 15 days from planting, and were also monitored for the whole duration of the study. The plant height was also taken in centimeter using a ruler at starting at 15 days, 30, and 45 days after planting. The stem diameter as well in centimeter was taken using this caliper, and leaf length and width were also taken using in centimeter using ruler at 15, 30, and 45 days after planting. As well as the number of leaves, the number and length of roots were taken at the number and length of roots were taken at 45 days after planting. And then the fresh weight were also taken up to at the termination of the study, 45 days after planting. And then this sample plants were dried at 60 to 80 degrees Celsius in an oven for 12 hours. And where the dry weight was taken using a weighing scale. So the result of the study, we have the first data on the percentage of survival of tissue cultured Cavendish banana plantlets. So this was taken after 15 days after planting, and just like any studies have conducted, 100% plantlet survival was observed in all treatments. So all plants were even alive up to the duration of the study. The second data on plant height, we have based on the result of the study, the stimulate combination with Bioforge and conventional practice significantly increased the plant height by 2 times from 15 days to 45 days after planting. So as you can see, treatment 7 has significantly, based on statistics, have highest plant height, which is also comparable to T4 or the stimulate and the conventional practice, and stimulate and bioforge combination. So this is now the growth of tissue cultured Cavendish banana plantlets from treatment 1 in the left. We have untreated, the treatment 2 are conventional practice, treatment 3 are using stimulate foliar application, treatment 4 using stimulate and conventional practice combination, and treatment 5 bioforge and and conventional practice combination, and treatment 6, the simulate and bioforge combination, and the treatment 7, the simulate and bioforge and complete and conventional practice combination. So in terms of stem diameter, as shown in table 3, the highest stem diameter were observed among treatment 5, treatment 6, and treatment 7, and also 4. So they has from 30 days to 45 days after planting, they have the highest. They are all comparable with the highest stem diameter. And in terms of length of leaves, the stimulate, again, treatment 7, stimulate and bioforge and conventional practice significantly increase leaf length by 4 times compared to the control. So as you can see on the table, the it has the highest length of leaves, which is comparable to the treatment 6 and treatment 5 and treatment 4 from 15 days to 45 days after planting. So again, in terms of width of leaves, the treatment 7, simulate and bioforge and conventional practice combination significantly increased the width of leaves by 7 times. So as you can see on the table, we have treatment 7, which is highest and are comparable to, again, treatment 4, 5, 6. And in terms of number of leads, the treatment 7 again significantly increased the number of leads by 50%, and also comparable to treatment 6, treatment 5, and treatment 4 as shown on the table at 15 days and 40 up to 45 days after planting. And in terms of number of roots, the stimulate and bioforge and conventional practice significantly increased number of roots by 80%. So as shown on table 7, treatment 7 is also comparable to T6, T5, and T4, T3, and T2, but and is compared to significantly different or higher compared to the control. So this is the effects of stimulate on the root and shoot growths of the tissue cultured Cavendish banana plantlets. So we have treatment 1 from the left, treatment 2, 3, 4, 5, 6, and 7. So in terms of root length, as shown in table 8, the treatments did not affect the root length. So statistically, they are all the same. And in terms of fresh weight, the tissue cultured Cavendish banana plantlets was increased by treatment 7 3 times. So it is also shown on table 9. It is also comparable to treatment 6, 5, and 4. And in terms of dry weight, the treatment 7 again increased the dry weight by 33 times and are also comparable to treatment 4, 5, 6 as shown on table 10. So this is now the effects of stimulate on the dry weight of tissue cultured Cavendish banana plantlets from treatment 1, 2, 3, 4, 5, 6, and 7. So to conclude, the combination of stimulate and bioforge with conventional practice increased the growth performance of the Cavendish banana plantlets, increasing plant height by 2 times, leaf length by 4 times, width of leaves by 7 times, number of leaves by 50%, and number of roots by 80%, fresh weight by 3 times, and dry weight by 33 times. And it is therefore highly recommended for tissue culture nursery practice, Cavendish banana plantlets nursery practice. So thank you very much for listening, and you are very welcome for your questions and clarification.